Hello everyone. In this video, we are going to learn about tips and tricks to identify the general histology slides. I have framed a systematic approach with the help of flowchart to identify all the general histology slides. In the general histology, we are having cartilage, bones, muscles, blood vessels, nervous tissue, lymphatic tissue and few connective tissue also. So in this video I'm going to take you through a flow chart, that is a one systematic approach to identify the general histology slides. So first we'll start with the slide which is having colorless images and HN is stained on. So in the general histology, the colorless one, that is the black and white, is the bone, TS and LS of bone, and in some places we are having the LS of peripheral nerve also as an unstained slide. So as I already said, this is colorless or something like a black and white image. It is very easy to identify this in the spotter because all other slides are stained with hematoxin and eosin. Here we'll get the plain black and white colorless image or few yellowish color in between. So as I already said, these two are the slides of bone. Here you can able to see the central Haversian canal surrounded by a concentric laminae. It is a circumferential laminae. So this type of arrangement seen in the transverse section of bone. If it is a longitudinal section, we'll get the very lengthy Haversian canal and interconnecting Boltzmann canal. So I hope you can able to differentiate between these two. In transverse section, you will get the circumferential lamina and Haversian canal will be in like a dot. In the longitudinal section, Haversian canal are longitudinally oriented. Moving to the next slide. This is the other black and white colorless image. This is actually the transverse section of nerve which is unstained and fixed with the osmium tetroxide fixative. That's why the myelin sheath around the nerve are stained black in color and the axon will be remain unstained. This is only available in few places. So we have seen all the black and white slides. Then coming to the HNE stained, hematoxin eosin stained slides, in that we can categorize firstly the slide which is having the lumen in it and without lumen. In the general histology slide, if it has a lumen, probably it will be of blood vessel. So here you can see the slides which is having the lumen either completely or partially. So as I already said, usually these are all the slides of blood vessels in which the blood vessels there are two types, arteries and veins. One way of differentiating arteries from vein, arteries usually having a regular lumen like this, these two, and veins will be having a kind of collapsed lumen. Then apart from that, if you observe the layers which is present, the layer which is close to the lumen is the tunica intima, then the next dark layer is the tunica media, then the third layer is the tunica adventitia. This is common for all the arteries and veins. In arteries, tunica media part will be more thicker when compared with the adventitia. See here, the tunica media part is more, here also. Here if you see, this is the lumen, this is the intima, then very thin tunica media, then very thick tunica adventitia. This you can appreciate even in the tenax also. So you can easily differentiate these two are the arteries and this is vein. Next we'll see how to differentiate large artery from medium size artery. Large artery is otherwise known as elastic artery. Medium artery is otherwise known as muscular artery. So as I already said, artery is having regular wall, okay, with the proper lumen. Here these are the two zoomed one I have taken. Here if you closely observe, close to the tunica intima, you can able to see a thick line that is known as interelastic lamina. Then in between the muscle fiber, you can able to see elastic fiber which is running. So elastic fibers is present randomly. Here in this one, the tunica intima is also not clear and you can't able to see very clear elastic fiber. This is because here completely the large amount of elastic fibers are present and very few smooth muscle. As there are more elastic fiber, we can't able to differentiate. But here elastic fibers are present in between the smooth muscles. So here even though elastic fibers are less, like an odd one out, we can able to visualize the elastic fiber here. So here we can't able to visualize the elastic fiber. So that's why this is the feature of muscular artery or medium size artery. This is the feature of elastic artery. Let's say in this image itself. Here in 10x itself you can able to see randomly oriented small elastic fiber. Here we can't able to appreciate, that's why this whole thing is the elastic fiber. So these two are the large artery and this is medium size or muscular artery. This is otherwise large artery is otherwise known as elastic artery. And one more clue to identify the large and medium size artery. In most of the slide you can able to appreciate the lumen completely or almost in the medium size artery. As the artery here the lumen is larger, you can't able to appreciate the whole lumen in 10x power. Then coming to the vein, we have already seen why it is vein. Irregular lumen, tunica media is very thin, tunica adventitia is more. Actually this is the slide of medium sized vein, and even though this is not having lumen, this is left over the blood vessel which is the large vein. How you can identify the large vein? As the large vein is completely collapsed, usually you won't get the luminal structure. It will be like a thin walled layer. Here this is the actually luminal side. Here the tunica intima, close to that tunica media is there. Then this whole thing is the tunica adventitia. That is the feature of vein. Tunica adventitia will be moved, but here within the tunica adventitia you can able to see darkly stained region, that is actually the smooth muscle. So smooth muscle presence in the tunica adventitia is one of the important feature to identify the large vein. In other places only the tunica media will be having the smooth muscle fiber. So these are the same points you can write for the slide identification also in your spotters. So till now we have seen the slides which is having the lumen, that is the blood vessel. Then slides without lumen are, first thing is the lymphatic tissue and other tissue. So why I am categorizing lymphatic tissue? Just by looking into the field you can identify the lymphatic tissue very clearly, because these are the very darkly stained slide one, and you will be having a lymphatic aggregation, okay, in a circle form or in the spleen, whether it is a red bulb white bulb distinguished, you will be having a lymphoid aggregation very clearly in the first look itself. So once you identify this is a lymphatic tissue, that is by seeing the lymphatic aggregation we'll identify this is a lymphatic tissue, within which we have to look for whether the slide is having stratified squamous non-keratinized epithelium or not. If it is there, then there is only one answer. The slide is the tonsil. If instead of the stratified squamous non-keratinized epithelium, if we are having Husserl's corpuscles and the slide is looking like an incomplete lobulation, then this is the thymus. If the lymphatic aggregations are divided into cortex and medulla, and cortex having more follicles with subcapsular sinus. Okay, so behind the capsule there is a sinus, that is a very thin space. If it is there, then it is of slide of lymph node. Then if these features are not there and it having the capsule, trabeculae, red pulp and white pulp, then this is the slide of spleen. So I will show you the individual slide in the upcoming slides. See, this is the lymphoid aggregation. Okay, either you will get slide like this or this view. Okay, here these are the lymphoid aggregation. Then in addition to that, see, these red arrows are showing the areas of epithelium, that is the stratified squamous non-keratinized epithelium. Okay, so as this is the tonsil, combination of these two, in the oral cavity you may have some submucosal glands also. These are the mucous glands. Okay, so the key point to identify the tonsil is the stratified squamous non-keratinized epithelium with the lymphoid aggregation. In most of the slide you may able to see tonsillar crypts also. Then here in this slide you can able to see the lymphoid aggregation. This is the cortex. Actually this is a low power. You can able to see in the cortex there are these lymphatic follicles are there. In the medulla these are the cords of lymphocytes. Okay, here this is the cortex and this is the medulla intendex. Here just below the capsule, okay, here also you can able to appreciate this is the capsule. Just below the capsule there is a thin space. Here you can appreciate clearly. This is known as the subcapsular sinus. This is an unique feature about the lymph node. Then in this lymphatic tissue you can able to see lobes. Okay, till now we haven't seen the lobes like this. This is the lobe over, this is the tenax. Here you can able to see there are some lobes which is not the completely divided. This is an incomplete lobe. Okay, here all these incomplete lobes are having a common medulla. This is the cortex and here the medulla. So in the center of medulla, if you closely observe, you may able to get few onion peel like appearance. Okay, cross section of onion like appearance. This is known as the Husserl's corpuscles. So this incomplete lobe with Husserl's corpuscles, this is the feature of thymus. Then the last lymphatic tissue. Here we can able to see this. This is a bit faded slide. Okay, I have used medium quality slide so that you can able to identify even if the slides are bit. Here if you closely observe, okay, this there is a capsule. These are the trabeculae, and what is the thing you can see very clearly is the difference patchy area, difference patchy area. Okay, so these are the areas of white pulp, and the other background is the red pulp. Within the white pulp, you may able to appreciate some central arteriole. Okay, eccentric arteriole you can able to appreciate. So this capsule, trabeculae, red pulp, white pulp, central arteriole, these are the features of spleen. So thus we complete the lymphatic system. So coming to the same flow chart, we have first seen the colorless H&E in the colorless bones, and in the H&E first we'll see the with lumen, without lumen. Then without lumen, first we completed the lymphatic system. So the remaining other tissues are cartilage, muscular tissue, nervous tissue, then connective tissue. So we'll see one by one. So when we see the cartilage, we broadly divide it into cartilage which is having the perichondrium and cartilage which doesn't have the perichondrium. So in the cartilage with perichondrium, if the cells are arranged in a cell nest, okay, that is the adjacent flattened surface, and if you see the matrix of the cartilage, it has homogeneous ground glass appearance, then the identification is the hyaline cartilage. If the cells are not in the cell nest and in between the cells we can able to see some darkly stained elastic fiber, then this is the elastic cartilage. If the perichondrium is absent, then there is only that is a white fibrocartilage in which bundle of collagen fibers and row of rounded cells, that is the chondrocytes, are present. That is the white fibrocartilage. So say this here, actually this is a slide of trachea. In the general histology, they will try to focus only this region like this. Okay, so the outer side you can see this is the outer fibrous and inner cellular layer of perichondrium. This complete thing is the perichondrium. Then if you move in, the background what you are seeing is the matrix which is ground glass like appearance, that opaque. Okay, you can't able to see any fiber at all. Okay, then in between the cells are the cells, if you closely observe, the adjacent surface of the cell are flattened. Okay, so this is a kind of cell nest. Okay, so this is the unique feature of hyaline cartilage. Then the elastic. Okay, here also the perichondrium is present. Okay, then the cells are here freely placed. Okay, there are a lot of space, so the cells are freely spaced. In between the cells you can able to see bit darkly stained area, those are the elastic fiber. This is the slide under the microscope. Here you can able to see cells are the in between, some darkly stained region are there, those are the elastic fiber. Here the perichondrium. So this is a slide of elastic cartilage. So this is zoomed one. Then coming to the third, that is the white fibrocartilage. Here the bundle of collagen fiber will be there. In between the collagen fiber, rounded cells with the rounded nucleus. So that is the differentiating feature from the white fibrocartilage and the dense regular connective tissue. If it is a dense regular connective tissue, these cells won't be having the rounded nucleus. Okay, so usually you may not be able to appreciate the nucleus, but you can able to see the bundle of collagen fiber running close to each other without any space in between. Then coming to the muscles. So you all know skeletal muscle is the muscle which is having the striated appearance. But in 10x, you hardly able to appreciate the striated appearance. What you can able to appreciate clearly is the long cylindrical unbranched muscle fiber. Okay, so in which peripheral nucleus are present. So this is the feature of skeletal muscle under 10x. If you go for higher power, you can able to appreciate the striation. Then moving to the cardiac muscle, in which you can able to see the branching muscle fiber. Okay, so that is the difference between the skeletal and cardiac. Cardiac, you can able to see the branching muscle fiber. In few slides you can able to appreciate these dark lines. Okay, what are these dark lines? Okay, so these dark lines are the intercalated disc. Then here the nucleus are located in the center of cell. Nucleus are located in the center of cell. So each cardiac cell will be having a single nucleus, unlike skeletal muscle which is like a syncytium. So single muscle fiber will be having more number of nucleus in the skeletal, but in the cardiac it will be a single nucleus. And few slide you can able to make out the perinuclear halo. Around the nucleus there is a white halo region which is an actual artifact. Then moving to the third muscle, that is the smooth muscle. So smooth muscle, in most of the places there won't be any separate slide. It will be a part of GI slide. So in that you will be having a spindle shaped cells. Okay, you can't able to appreciate the shape, but each and everything is a separate cell. Within that you will be having a flattened nucleus. So this is the feature of smooth muscle. Then we'll see the nervous tissue. So first thing we'll start with the optic nerve. So this is the transverse section of optic nerve in 10x. When we say this as an optic nerve, you have to visualize the central retinal artery and central retinal vein. Okay, so what are all these bundle? These bundle are the bundle of axons. Okay, so this completely forms a nerve. So this is the feature of all the peripheral nerve also. But the unique feature in the optic nerve, which has the blood vessels, okay, artery and vein in the center of the nerve bundle. So this is the optic nerve. If this is not there, then we can call this slide as a peripheral nerve. So even in some colleges, peripheral nerve slide is also available. So as I already said, this is a transverse section. So if you see the optic nerve slide in the low power, you can able to appreciate the covering, that is the meningeal covering. So this optic nerve will be having the meningeal covering, as this is derived from the brain. Moving to the next, these two are the bit confusing slide in the general histology. Actually, these are the slide of sympathetic ganglion and sensory ganglion, in which you can able to see the cell bodies. These are the cell bodies and these are the cell bodies. So the difference between these two, in the sensory ganglion, you can able to appreciate cell bodies are arranged in groups. Here one group, here one group. In between, there will be axons. Fibers are running. In the sympathetic, these are randomly placed, not in the groups. Then one more easy differentiating point, you can able to count the number of cell bodies. That is, the cell body is quite larger, so the number of cell body within a field will be minimal. So you can actually able to count. But here in the sympathetic, as the cell body is smaller, you can't able to count. There are numerous cell body present in a single 10x field. Hope you can able to differentiate these two based on the arrangement and size of the cell body. So the next thing, we'll see the connective tissue, that is only the adipose tissue, and dense regular we have seen with the white fibrous cartilage. So in the adipose tissue, this is a cell which contain a single large vesicle of fat. So all the cytoplasmic content including the cell organelle pushed to the periphery. So you can able to see these are the nucleus in the periphery. So it's like a signet ring appearance. Okay, so this is the classical feature of adipose tissue. So coming to the summary, so in the general histology slide, we have first divided into colorless and Hutchins slide. Colorless or bone, rarely the LS of nerve which is fixed with the osmium tetroxide. Then Hutchins slides with lumen, without lumen. Without lumen, the tissues are lymphatic tissue and other tissue. With lumen, it is usually the blood vessel. And we have seen that what are the differentiating feature in the lymphatic tissue. Then in other tissue, we have seen the 3 cartilage, 3 muscles, nervous tissue and connective tissue. Hope this video is helpful for you. So thanks for watching. Hit the like button and you can ask any doubt in the comment section. If you want any further video, subscribe to our channel and mention what are the anatomy videos you want to see in our channel in future in the comment section. Thank you.